Okay, so welcome everyone again to the Green Ear Hub webinar series. My name is Barry Crosby and I'm one of the communication officers for the Green Ear Hub project. If you've been here with us before, you'll know that the Green Ear Hub project officially started in September 2022 as a coordination and support action of the European Commission under the Horizon Europe programme. And it currently represents 15 EU ERANETs, co-funds and self-sustained initiatives in the field of agri-food and biotechnology. We started these breakfast clubs in October 2023, and we've had 5 very successful series so far. The first was Circularity in Agriculture. The second series was related to biotechnology to advance sustainable solutions for bioeconomy. The third was Sustainable Food Systems. The fourth looked at precision agriculture technologies for a sustainable future. And the one that we just finished last month was on plant health and protection. If you're interested in listening to the talks related to any of the topics from the series just mentioned, please do note that our recordings are now available to view on the Green Era Hub YouTube channel. Today, we're going to continue our new Breakfast Club webinar series, which focuses on the topic of animal health and welfare. So for the next 2 or 3 months, we will host a series of expert-led discussions aimed at fostering a deeper understanding of sustainable strategies for improving animal health, preventing disease, managing pests, and strengthening the connection between animal, human, and environmental well-being. And just a few housekeeping announcements. Please do feel free to type your questions into the Q&A chat section, which I've enabled for everyone. I'm going to do my best to address as many questions as possible within the time that we have this morning, which is 1 hour. If at the end of the session you have a question for either of our speakers, please do remember to address the speaker in the question. Now, today we have 2 panelists joining us, Professor Romain Volmer and Professor Beatrice Prieto Simon. I will open our session this morning with Romain Volmer, a veterinary virologist specializing in infectious diseases, with a focus on avian influenza at the École Nationale Veterinary de Toulouse in France. An experienced researcher and lecturer in infectious diseases, Professor Volmer leads research on the pathogenicity and transmission of avian influenza and viruses. His work combines viral genomics and molecular biology to investigate how host species, particularly chickens and ducks, influence the emergence of highly pathogenic avian influenza strains. Supported by national research funding and the chair of biosecurity, his research aims to improve our understanding of virus evolution and host adaptation in the context of animal health and biosecurity. Today, Professor Volmer's talk is entitled Identification of Factors Driving the Emergence and Spread of Avian Influenza Viruses with Zoonotic Potential, which is part of the EU-funded International Coordination of Research on Infectious Animal Diseases initiative. Romain, thank you very much once again for accepting our invitation to this webinar series. We're absolutely delighted that you're able to join us this morning. I'm going to stop sharing my screen now so you can start sharing your presentation. Thank you very much.
So hello everyone. So I'm pleased to share some of our research and describe a little bit the project that was just mentioned. So I will start the presentation now. So the project that we will discuss is indeed funded by the ICRAD, so the consortium, the coordination of research on animal infectious diseases. And the project is called Flu Switch. The project is a multinational project with 8 partners. So I'm from Toulouse in France. We have partners from Germany, from the UK, from Italy, another one from the UK, from Scotland, We have partners from the Netherlands, from Poland, and from Turkey. So we built this consortium focusing on avian influenza virus to understand better the ecology and zoonotic potential of avian influenza viruses. So on this drawing, first you see a virus, so an influenza virus here, that has a genome that is made of RNA. It's a segmented genome that has 8 segments that are inside the virus particle here. And what is important to know about this avian influenza virus is that because of their genome structure and nature of the RNA virus, they have a high potential for evolution. And this is really the major question of importance for both animal health, because some of these viruses can cause disease in animals either in the wild or in domestic animals, causing economic losses and health problems, so comprising animal welfare. But one of the major concerns, and that is very timely, is their zoonotic potential. So their ability to cross species barrier and jump from an animal species to human. So in nature, avian influenza virus exists mainly in water birds, like seagulls or ducks or geese, where they replicate without causing major clinical signs. They can then be transmitted occasionally to domestic animals, like poultry, and mostly they cause few symptoms, but sometimes they evolve from what we call low pathogenicity influenza virus to high pathogenicity influenza virus. In this case, the virus will cause in birds very severe disease, usually leading to the death of animals and sometimes of the entire flock within a few days. So first of all, a big distinction in the field of avian influenza virus virology is the distinction between low pathogenic viruses and highly pathogenic viruses. And we hypothesized in this project that the high pathogenicity influenza virus could have an increased zoonotic potential. And their increased zoonotic potential is probably linked to the fact that when a high pathogenicity virus infects a bird, it spreads more rapidly, it is excreted at higher levels. And being excreted at higher levels means it will be more likely to infect other animal species, including mammals. So on this drawing, you can see some mammalian species. So swine have been infected and have been shown at least once to lead to emergence of a pandemic virus in the past. But since the major spread of H5N1 high pathogenicity virus around the globe, we have seen many cases of mammalian infections in sea mammals, in foxes, and of course, since more than 1 year, in cows, especially in the US. When the virus replicates in these mammalian species, it may further adapt to humans and this may lead to emergence of a new pandemic virus. So for the project itself, we focused on the initial steps of this evolution process, and more precisely on what's happening in birds and what are the factors that will lead to the evolution from low pathogenicity virus to high pathogenicity virus. So the questions of the flu switch project are listed here. So we are investigating the virological factors that influence the emergence of high pathogenicity avian influenza virus. So by virological factors, we mean what's inside their genome might predispose them to evolve to high pathogenicity virus. We're also looking at host factors. For example, we are investigating whether the immune response in some animals might influence this evolution. And of course, we are looking at the factors that influence the spread of this virus and mammalian transmission. So this is a big project, and during this presentation, I'm going to focus on a specific aspect of the first question, which is what are the virological factors modulating the emergence of high pathogenicity avian influenza virus? So first, I need to go to a more precise definition of what we call low pathogenicity and high pathogenicity avian influenza virus. So on the left, you have a low pathogenicity avian influenza virus, and the main determinant of a change of a switch from low path to high path is located in the hemagglutinin of the virus, which is a glycoprotein that enables the virus to enter cells. In low pathogenicity virus, you have a monobasic cleavage site in the HA. And this monobasic cleavage site is recognized by trypsin-like proteases that cleave the protein, the precursor, in two parts, HA1 and HA2. This cleavage is required for the virus to be able to enter the cells. If it's not cleaved, the virus is not able to enter cells. For a low path here on the left, the virus in birds will replicate in the respiratory tract and digestive tract. And the reason for this tropism is not because of the distribution of receptors, it's because of the distribution of the proteases that cleave the HA. So trypsin-like proteases are located in the digestive tract and in the respiratory tract. And therefore, because the tropism is limited, the symptoms are mainly limited. And then also the level of virus replication is more limited. When the virus evolves to a high path, the main determinant is the cleavage site, is the evolution of the cleavage site of the HA. The HA cleavage site becomes what we call polybasic, which means that there are more amino acids present in the cleavage site, and therefore this cleavage site will be recognized by furin-like proteases that are expressed in all cells of the body. And because the HA can be cleaved in any cells of the body, the virus is able to enter any cell of the body. Therefore, the virus is able to spread what we call systematically, which means it will be found everywhere, including, for example, in the brain of the animal, and will cause very severe disease. So the main focus of my research currently is to understand what causes this evolution at the molecular level. So how do these basic amino acids, how are these basic amino acids introduced? And this is what I'm going to describe. So I'm going to describe some results of research on this specific topic. So our goal is to identify the mechanisms that lead to the evolution of this cleavage site. And we and others hypothesize that there might be some specific features in the RNA, so in the genome, in the gene encoding this HA, so the hemagglutinin, that may predispose some viruses to evolve to high path viruses. And it could be either the primary nucleotide sequence, that is pictured here, that might influence this. Others have proposed that some big structures formed in the RNA could influence this evolution. And more recently, some others have proposed that transient secondary structures formed in the polymerase, outside of the polymerase, might influence this evolution. So on this drawing, you see how the replication occurs. You have in black what is called the template RNA, and in blue, the newly synthesized RNA. And in this, the replication, the copy of this RNA occurs in the viral RNA-dependent RNA polymerase of the influenza virus. So I'm going to present you first how we study this. So which tools we developed. So we used what we call reverse genetic to genetically modify an influenza virus, to study this in a safe way and efficient way. We created a virus that has a transgenic HA expressed after another gene that's called PA. And this transgenic HA is the one we want to study. And our goal was to have a virus that carries a transgenic HA so that it is being replicated by the viral polymerase, but with no function associated with this transgenic HA. So therefore, we put this transgenic HA out of frame. We introduce stop codons to make sure that this HA is not translated. So it will not be part of the virus structure itself. And the function of the normal HA is carried by the red here, HA that is present, the normal one in the virus. So the goal here is really to have a system where the HA will be replicated and in which we can introduce any mutation to study the link between sequence and insertion events. To study this, we need an accurate and sensitive sequencing method, and I will not go into the detail, although you may have questions that I would be happy to answer on this. And we detect very rare events, so at a level very low frequency. Because of this method, we are confident that this method is able to detect this type of events. So we compared two viruses first. Here, the wild-type virus on the left, and a mutant that has just two substitutions, that's called 24A2B here. And what you can already appreciate from this graph is that the virus on the right has many more insertions in the HA cleavage sites that are depicted here. On the y-axis is the frequency of insertions, and here is the place where they are introduced. We decided to test whether these structures could be involved, and we modified the sequence here and here to destroy these structures that are present here. And we have no effect of these modifications. Therefore, we say, first of all, that the nucleotide sequence has an influence on the insertion rate, but no impact of transient RNA structure on this phenomenon. We next compare different viruses, and I will go quickly here, just to tell you that we compared 4 more mutants. And what we conclude from all this analysis is that the main determinant of insertion is not the big structures, nor the transient structures here, but the primary sequence. And as soon as you have a stretch of at least 6 A's in a row, you have a high level of insertions that are represented here by the gray bars. We next investigated natural H5 emergent events, so that have been described. And here on the top, you have the low pad virus, and here the high pad. And you can see that the cleavage site is more basic and increased in size. And we hypothesize that these viruses may have emerged because of an intermediate virus that has 6 A's in a row. The normal LP, low pathogenic virus, doesn't have this. So we created these viruses and analyzed their insertion rates. And you can see that as soon as we introduce 6 A's in a row, we have a high level of insertions that may lead to acquisition of a polybasic cleavage site. We think that these insertions are caused by polymerase backtracking and reannealing. So this is the catalytic site of the polymerase. And here in black, you have the template, and on the top line, the newly synthesized RNA. So here you have perfect interaction complementarity, but if this complementarity is weak in terms of thermodynamic, the product template might detach and move and realigns if the thermodynamic stability of a newly aligned product template is stable enough. So here in this case, it went back 3 nucleotides, was able to realign because the stability is quite good. And because it has gone back 3 nucleotides, it will copy this sequence twice. So CUU has been copied twice. And this is actually the insertion we detect here. CUU is translated as a complementary GAA in our analysis. So everything here is due to the thermodynamic stability of a product template and based on the sequence at this specific position. So to conclude this presentation that is described just a specific part of the Flu Switch project, so the one that I'm that we are really doing in my lab, we conclude that these insertions require a stretch of 6 A's at least that are not very frequently detected in low pathogenicity virus in nature. And the reason why they are not frequently detected is because we think that they might quickly evolve to a high path because of the high level of insertions that they cause. And when we have more insertions in this cleavage site, the newly generated virus will have a major selective advantage because it's highly pathogenic, replicates to much higher level than its precursor, and therefore evolve to a high path virus with more insertion with a bigger cleavage site. What we are doing now is to model this process to try to, so using I would say some basic mathematical modeling to try to understand, try to see if we can really predict the risk of insertion based on the primary sequence of the cleavage site. And we have obtained some nice results in this, and we are currently using it to identify H5 sequences that would be at higher risk of evolution to high path based on the primary sequence. So I thank you for your attention, and I'm happy to answer any questions.
Thank you very much for such an interesting and insightful presentation, Roman. I just want to remind everybody that the chat box and Q&A section is open, so please do feel free to type a question for Professor Vollmer at any point. Roman, just I was kind of really struck, I think, by your presentation. It was an excellent presentation, examining the factors driving the emergence and spread of avian influenza viruses with zoonotic potential, particularly at the kind of molecular level. I would like to ask, if I may, about the kind of ecological and environmental factors driving the emergence and spread of avian influenza viruses with zoonotic potential. And I was just wondering about environmental conditions. So, for example, climate or agricultural practices. Have any of these shown to favor the emergence, in your experience, of highly pathogenic strains of viruses?
So, first of all, we need to define what we call emergence. And there are different definitions. So, emergence of a high path virus at the molecular level is what I'm studying here. So it means that you have introduction of nucleotides in the cleavage site. And this is something that happened at least 50 times in history. And it's quite rare. 50 times is not a lot. But each time, it caused outbreaks. And that's why we know of them. But most of the time, these outbreaks were controlled, and the virus disappeared, was not able to spread to other animals. What we're dealing at the moment is a specific situation linked to the H5N1 virus that emerged in 1996. And in this case, the emergence event is not well known. We don't know exactly precisely where it came from, what was the precursor. But we know that it's a specific virus because it has been able to spread now globally, including, for example, in the Antarctica. And one of the reasons of this, I would say, success for a virus, not for us, but for a virus, is because it has been able to exchange gene segments with a lot of other viruses that infect birds. And one specific event has been described, which was the recombination, so the exchange of gene segments with viruses that circulated in girls. And this probably happened most likely in Europe. And the new virus that emerged from this was able to infect very efficiently wild bird species and was able then to extend its host tropism. And because of this, it was then able to also stay longer at certain places. For example, the virus lost its seasonality. Normally in Europe it was winter times. Now, at least in 2024, 22, sorry, in the last years it was less seasonal. So I would say that the impact, for example, of climate may have played a role because some bird species that were infected with other viruses probably could have stayed longer in certain regions, for example, in Europe. And because these viruses harbored some specific viruses, this may have increased the likelihood of gene exchange with H5N1 viruses. So, in this sense, I would say that indeed environmental factors can play a role, and one of the major impact is on the migration process of these wild birds, so that could influence the whole process and have major consequences. Yes.
Thanks very much, Romain. And just to follow on, I suppose, to that, you mentioned about migratory bird patterns and how that has a tremendous impact on the global spread of avian influenza viruses. I was just wondering, thinking back to, I suppose, the outbreak of coronavirus, I was wondering about the role of maybe wetlands, live bird markets, that kind of thing, in terms of sustaining and amplifying avian influenza reservoirs. Would you be able to maybe tell us a little bit about that and how significant they
Yes.
are in terms of?
Well, regarding wetlands, I think that the link is direct because, as I said, water birds are the reservoir of avian influenza virus, and therefore you find a higher diversity of viruses in these wetlands. So they can be a source of new viruses, either because they just emerge from these wild birds, or because, as I just mentioned, these viruses mix with other circulating strains in the process that is called reassortment. That is the exchange of segments between two influenza viruses that co-infect a host and actually even co-infect one cell within a host. So in this case, you can have a new virus that emerge. So they have a role, wetlands have a clear role in maintaining the virus and in generating diversity. Then regarding live bird markets, I'm not personally investigating this, and we are not investigating this in the project, but I have some close collaborators who are investigating this. And one major observation that is really reproducible regarding live bird markets is again the diversity of viruses that you find and the number of samples that for you, that which you analyze that are positive for avian influenza virus. I have a colleague, Claire Guinard, who's working in Toulouse, and she's working on the investigation of influenza virus prevalence in live bird markets in Cambodia. And you have major differences between the rate of positivity of farms compared to live bird markets. So something like 5 to 6 times more viruses present in live bird markets compared to farms in terms of frequency. And the reason is that live bird markets become a node where positive farms can virus can be transported, and where because there are live bird markets, animals can become infected, and therefore it's really hot. Hot hub for virus detection and maybe also for virus evolution.
Fascinating stuff. Roman, just sorry, one last question just before we wrap up, and it relates to epidemiology and surveillance. Now, again, this may fall outside of the scope of your research project, but I was just wondering, based on your experience, what are the most effective strategies for early detection and surveillance of avian influenza with zoonotic potential?
Well, I mean, first of all, we can rely on of course molecular detection, which is very sensitive. I would say that now a lot of people are developing methods to sample the environment, and this is something that is very powerful because you can have at one point traces of virus that you actually that can come from many places. So it's maybe more efficient than sampling, many farms. So if you have the ability to have a certain sampling method that enables you to detect possible infection from many sources, like because the water flows from different farms into a specific pond, let's say like this, just this simple image, you may be able to detect using one sample the presence of virus coming from many farms. And if you detect it, then you go to a different, you move and you try to see where it comes from. So I would say that indeed environmental sampling is something very efficient, but it's also very linked to the technology. So very linked to the sequencing methods that need to be very sensitive and very accurate. So there is still needs some tools for improvement regarding this, but already it's very powerful. So I think indeed, I would say that this type of sampling really complements what is being done at the farm level, where you can also swab animals, but also swab the environment, let's say dust or something like this that you can collect, which is also very can be very sensitive to detect a new virus. Yeah.
Fascinating stuff. Roman, thank you very much for your excellent presentation. I think it's really a kind of a tremendous overview of the project, and thank you so much for answering the questions at the end. Now joining us is Professor Beatriz Prieto Simon, ICRE Research Professor at the Catalan Institute of Chemical Research in Spain, where she works as an analytical and materials chemist, specializing in biosensing, nanotechnology, and advanced functional materials. Holding a PhD in chemistry, Professor Prieto Simon has conducted research across multiple international centers in Europe, Asia, and Oceania, focusing on the development of innovative biosensor technologies. Her work bridges chemistry, biology, and materials science to engineer responsive platforms for diagnostics and environmental monitoring. She currently leads the Bio-Inspired Nanotechnologies Lab, where her team develops cutting-edge materials for biosensing applications. Professor Prieto Simon's interdisciplinary approach aims to create sustainable, sensitive, and real-world ready sensing systems inspired by natural processes. Today, Professor Prieto Simon's talk is entitled Tanno-based biosensors to support a precision agriculture approach for improved bovine mastitis management, which is also part of the EU-funded ICRAD initiative. Patrice, thank you very much again for accepting our invitation to the webinar series. We're also absolutely delighted to have you joining us today. So without further ado, I'm just going to open up the floor to you. I'm going to stop sharing my screen so you can start sharing your presentation. Thank you very much.
Thank you very much for your kind introduction. So first I'm going to share the slides. So, well, good morning. And first of all, I would like to thank the Green Air Hub for the invitation to share our research journey throughout the Biosensible Precision Mastitis project supported by ICRAD, by the ICRAD initiative. In a nutshell, the aim of the project was to deliver diagnostic solutions to improve management of bovine mastitis. And indeed, this is very well aligned with the research we perform in our laboratory that mostly aims at developing the next generation of diagnostic tools to support the prompt diagnosis of infectious diseases, the guidance of the therapy required, and also reducing the spread of antimicrobial resistance. Pivotal to the success of the project was our consortium that consisted of the Riga Stradiņš University in Latvia, with experience studying the animal immune response, the Research and Innovation Center Pro Academia in Poland, with access to cattle farms and microbiological facilities and with the skills to analyze milk, Sequomics Biotechnology in Hungary, with experience in genomics and bioinformatics, and our research group first at the Universitat Rovira i Virgili and currently at the Institute of Chemical Research of Catalonia, where we have experience in the design, nano and microfabrication and surface engineering of advanced materials to be integrated into diagnostic tools based on biosensing principles. So the overarching aim of the project was to develop new tools for the early diagnosis of cows infected with bovine mastitis. And why bovine mastitis? So because bovine mastitis is one of the three main issues affecting dairy cows. It's mostly caused by a bacterial infection, and it can manifest both clinical and subclinical states, the later ones being very concerning because there are no clear symptoms. Bovine mastitis causes others inflammation and pain and discomfort to the cows and also induces physical and chemical changes of milk. The impact of bovine mastitis is huge. And the economic burden is enormous, and this is related to the reduction in milk yield, milk quality, and also related to the treatment cost involved. Also, what is really concerning is that many times there is a late and poor intervention. And for instance, in the cases that there are signs that infection can be controlled, then antibiotics are administered and the isolation of cows is enforced. The problem is that many times antibiotics are administered without clear evidence, and this, of course, contributes to antibiotics overuse that not only has an effect on the animal's immune response, but also can end up with antibiotic residues in milk or residues released in water and soil, again contributing to the emergence of antimicrobial resistance that is a very highly concerning public health issue worldwide. So for us, it was clear that by providing prompt diagnosis linked to prompt intervention that could help in the better management of bovine mastitis. But the problem is that the diagnosis of this infection faces several limitations. So for instance, for clinical cases, mostly the diagnosis is based on visual inspection when farmers observe changes in the quality consistency of milk, if there are clots in milk, or also when they observe that the cow's udder is swollen or red. On farm, somatic cell count is commonly used as an indicator of the likeliness of milk to contain harmful bacteria, but this can only be used as to identify potential cases. In lab, it's true that we have cell culture and PCR that are very good techniques to confirm infection and also identify the causative pathogen, but these are time-consuming and costly techniques. And more important, none of those techniques can ensure the diagnosis at the early stages of the disease. That is when animals are highly infective and therapies are most effective. So that's why the rationale of our project was to ensure that we could deliver tools to support the early and efficient on-farm diagnosis of bovine mastitis. To do that, we mainly focus on three innovations. The first one was targeting host immune response-derived biomarkers at those very early stages of the infectious process. The second one was leveraging the presence of these biomarkers in milk to allow non-invasive and stress-free diagnosis. And finally, the third one was harnessing advances in sensing technology to later ensure or to facilitate technology adoption. In the end, as a result, what we get is a paradigm shifting concept that mostly focuses on confirming infection rather than just pathogen exposure, as differently to what most diagnostic techniques do that are based on the detection of biomarkers related to the presence of pathogens. So the four premises of our research were, as already mentioned, the identification of a distinct biomarker signature, including microRNA, cytokines, and antimicrobial peptides as those host immune-derived biomarkers I mentioned before, to enable action before pathogen replication and manifestation of symptoms. The second one was targeting mastitis in a natural setting. Instead of tackling controlled intramammary infections, instead of using animal models, we prefer to work in a natural environment to avoid underestimating the potential effects of mastitis acquired through natural patterns of pathogen exposure. Third, we focus, as I mentioned before, on harnessing advances in biosensing technology to ensure that we could provide a label-free and direct detection of the firstly identified biomarkers. And finally, to integrate those sensors into a point-of-care device, easy to operate on farm and enabling high-frequency readings. So for all that, as already mentioned at the beginning of my presentation, the network of researchers in our consortium was essential to provide that multidisciplinary approach that goes from animal health to technology translation. So I'm going to share with you a little bit of the results of this project, going through all the different steps. We started with the identification of these host immune-derived biomarkers, and to do that, we had to collect milk samples. We did that during 5 different rounds, and we collected milk from a herd of Holstein Friesian dairy cows. And to classify those milk samples into healthy, clinical, and subclinical, we used somatic cell count values, and also for clinical cases, of course, additional symptoms observed. On the one hand, with these milk samples, the first thing we did was to extract RNA that was purified and then used to prepare microRNA in vitro libraries that were sequenced, and the results were analyzed to identify a unique microRNA pattern. What we observed was that a panel of different microRNAs were consistently overexpressed along the 5 rounds of collection of milk samples. And in the end, we chose microRNA 223, 221, and 146V to continue with the development of sensors. On the other hand, we performed some milk smears in histochemically staining using different factors to select a panel of cytokines and antimicrobial peptides as potential biomarkers of bovine mastitis. In the end, we found a significant decrease in the expression levels of interleukin-10, beta-defensin 3, and beta-defensin 2 in the milk from infected cows. So we choose to continue in the development of our sensors targeting these 3 biomarkers. In parallel, we were interested in finding a pathogen-specific biomarker signature, but unfortunately, we could not find any clear relationship between bacteria and the biomarkers. It is true that if we, in general terms, if these are the results for the last collection round, if we look at the values, there is a correlation between like high total bacterial count and high SCC levels, or higher levels of Streptococcus aureus and Staphylococcus aureus in infected samples, and also the presence of Escherichia coli and Streptococcus agalactiae exclusively in samples coming from infected cows. But in the end, that could not be used for diagnostic or prognostic purposes. But of course, we are aware, and we know this is really important, that this information is really valuable later to guide the antibiotic treatment. So moving to the biosensing technology we developed, I'm going to focus only on the results we obtained for the microRNA sensors. As already said, we were looking for a biosensing technology that could enable this label-free and direct detection of the biomarkers. And for that, we chose to work with materials that feature nanochannels, where we confine the recognition of the biomarker. So in this case, these nanochannels were modified with capture proof that is complementary to the target microRNAs in a way that when the microRNA hybridizes, this causes a partial blockage that can be directly, and as I said, label-free, measured via electrochemical means, either using voltammetric or impedimetric techniques. In parallel, though, I would like to share with you that we work with a simpler platform, more straightforward, just to confirm the diagnostic potential of the 3 microRNAs selected. In this case, we work with commercial gold electrodes, where we self-assemble a thiolated Redex target capture probe that is complementary to the target microRNAs. And the mechanism works in a way that when the microRNA hybridizes the mobilized capture probe, this causes a conformational change that alters the electron transfer kinetics. And this can be easily measured by changes of the voltammetric responses. So going back to the real sensors that we were aiming to develop with these nanochannel these materials that feature nanochannels, we work with 2 different materials, porous silicon and porous alumina, that are fabricated through electrochemical anodization. And after that, in the case of porous silicon, we had to go through a carbonization process to make these substrates conducive to electrochemical sensing. And on the porous alumina based biosensors, we work with porous alumina membranes that were placed on carbon electrodes. Then we proceeded with the chemical functionalization of these materials to later enable the covalent binding of the capture probe, as I said, complementary to the target microRNAs. In the case of porous silicon, after assessing different strategies, we found that the best results were achieved with the electrografting of hydrazide-based diazonium salts. And in the case of porous alumina, the best results were achieved through silanization with Aptis. Just to emphasize that the reason to use these type of structures is mainly, as I said, the possibility to have a label-free and direct detection, but also because by confining the binding event, we can greatly increase the sensitivity, and also we can mitigate matrix effects. And working with milk, which is a very challenging sample, that could help significantly. So all the studies performed, all the optimization steps were initially performed with DNA instead of RNA as our target. And this is mainly because it's cheaper, it is a lot more stable, it's a lot easier to work with DNA. So we perform all the optimization with DNA in buffer, and then we move to confirm that the system also works with RNA before testing or challenging our sensors with milk. With the porous silicon sensors, unfortunately, after trying several strategies, we could not mitigate matrix effects, even diluting milk 1,000 times. It was not possible. But fortunately, with our porous alumina sensors, we found that using with a pretreatment based on blocking the sensors initially with skim milk, then there was a significant difference between sensors and controls, enabling to proceed to challenge these sensors with milk. In parallel, just not to forget, so we're working with the other platform just to confirm the diagnostic potential of the 3 microRNAs. And we performed the experiments in the same way, starting with DNA, moving to RNA, and then challenging the sensors with milk. So this is just to show you how challenging it is to work, to develop sensors when working with milk. The first studies we performed were with fresh milk that we purchased from the supermarket. And while DNA was working really well, spiked DNA, when we moved to RNA, we lost more than half of the signal gain measure. When we moved to raw milk directly from farm, we completely lost the signal. So we tried different strategies. Dilution didn't work, and being aware that the main problem is caused by the presence of RNases in milk, we tried different strategies to denaturate RNases, like boiling milk, adding RNase inhibitors or proteinase K. And finally, we got some pretty decent results using proteinase K, but not reaching the levels of signal gain obtained with DNA. As a result, rather than using milk, as we said, again, to confirm the diagnostic value of the 3 microRNAs, instead of working with milk, we work with RNA extracts from the milk collected on farm. And the study here that I'm showing is to demonstrate the performance of the 3 microRNAs, and is based on plotting what is called the receiver operational characteristic curves. That basically what we plot is the true positive rate that is related to the sensitivity versus the false positive rate that is related to the specificity. And what is important here is the area and the curve. That is a kind of a quantitative measure of the diagnostic performance. When this number is close to 1, means that there is perfect accuracy, and when it goes close to 0.5, it means that there is no diagnostic value. So the results that I'm showing here confirm that microRNA 146b and 221 have strong diagnostic value, while 223 has a moderate diagnostic value. But what is really important here is that by combining 3 of them, by using a multi-biomarker detection strategy, we can greatly increase the diagnostic accuracy, meaning that we can reduce the likelihood of having false negatives, and also we can increase the overall reliability of this sensing approach. And just to finalize, I would like to show you the results we obtained with our pores alumina sensors. In this case, we analyzed 10 milk samples collected directly from farm. And the results here show that we can significantly discriminate the milk samples coming from healthy cows from those coming from cows infected with subclinical mastitis. We were very happy with those research results, but I would like to also emphasize that the project really focused from the beginning on maximizing the impact. And for that, we prioritize from the project inception, co-creation with end users, and also engagement with stakeholders. The feedback received from farmers and veterinarians was really useful to identify which are the requirements to later facilitate technology adoption. And also it was very interesting to engage with diagnostic providers and manufacturers, because what is clear is that for the next stage, we have to move to a prototype that can convince farmers that the cost benefit, that is really what they are looking, is positive. And also all the feedback received underscore the importance of harmonization. That is also something that we consider from the very beginning of the project, and that's why we try to compare all our results with microbiological analysis and immunological assays. So where we are so far. So we have a proof of concept of a sensor that can accurately discriminate milk samples from healthy cows from that coming from cows infected with bovine mastitis. And the next step, and indeed we are currently seeking actively for further support, would be the integration of these pores alumina sensors into a lab-on-a-chip device that later on can be implemented into existing milking infrastructure. And why that? Because the expected impact is huge. Not only we expect by using this device an increase farm profitability and productivity, but also this device can contribute to rational and informed use of antimicrobials, and also help producers to be better informed to manage endemic diseases. Here are some details about the project. We have a YouTube video that I invite you to have a look, that summarizes really well and briefly the aim of the project. And finally, I would like to acknowledge all the national funding agencies that supported the Biosensor for Precision Mastitis project, as well as ICRAT for all their support. And thank you very much for your attention. I'll be happy to answer any questions you might have. Thank you very much.
Beatrice, thank you very much for such an interesting and insightful presentation. Just want to remind everyone that the chat box and Q&A section is open. So if you have a question for Professor Prieto-Simon, please do type that in at any point between now and the end. Patrice, just while we're waiting for one or two questions to come in, I'd just like to ask you a little bit about the biosensor design and functionality, if you're able to speak to that just a little bit. And I was just wondering if you could point to the kind of core principles behind channel-based biosensor technologies, and maybe kind of describe those for us, and particularly how they maybe differ from kind of more traditional sensing methods.
So yeah, basically it's a very good question, and it's really related to the research we do. So basically we work with this type of materials that, as I said, are porous materials. We have worked with others apart from porous silicon and porous alumina. And basically the nanochannel based sensing mechanism relies on confining the binding event. So usually for electrochemical sensors, we work with planar electrodes. And moving to a confined environment, this means that upon recognition of the analyte by the immobilized bioreceptor, there is a partial blockage that is a combination of stereohedrons. So if we are targeting a virus, for instance, with the right pore size, this is going to cause a significant blockage of the pores, but also works with small molecules like the microRNAs that I was showing before, because then there are some electrostatic interactions that can play in our favor. So meaning that if we, in the solution, if we add a redox species that has the opposite, the same charge, then there is a repulsion, and also it's increasing that partial blockage. So it's, as I said, a combination of stereohedrons plus some electrostatic interactions. And with that, we can significantly increase the sensitivity, because we can nicely tune pore size to maximize that partial blockage. But in addition, we can also play with the size of the pores, do a kind of size selective exclusion of potential interferences. So this helps a lot when dealing with complex samples, such as in this case, milk.
Fascinating stuff. Patrice, I was just really interested also in you just mentioned towards the end of your presentation about the kind of uptake by farmers, by dairy producers. And I was just wondering about, I'm imagining that there would be certain challenges there in terms of uptake, in terms of encouraging farmers, dairy producers to kind of embrace these new technologies. And I'm wondering, can you maybe just tell us a little bit about that? Like, what do you foresee as being the kind of, I suppose, major challenges in deploying biosensors in field conditions on dairy farms, etc.?
It was very interesting. So we were not, there were interviews with farmers, and my team was not the one in charge of that, but of course with all farmers, we were sharing the results, and the feedback received was really interesting, because there are things like, we don't think that it's totally different if it's a very small farm or a huge farm. And while some of them are very open to innovation, the others are very reluctant to change anything that they are doing. And also perceiving the how important or how significant is the impact of bovine mastitis is also very different depending on from where it comes the feedback. But it was very useful in the sense that they told us, like, for instance, it has to involve the minimum additional labor possible. So if it's something that is not going to change much, I mean, it's not going to add more work to what they are doing. Most of them, they are happy with that. They also insisted that they don't want to change to apply major changes to milking infrastructure. So if the changes are minor, it's okay. But then other details like how is going to be to use them. So they were worried about receiving the right technical guidance. How to use the cost, of course, is one of the key aspects to take into account. We were suggesting something that can be changed every time a disposable sensor, and yeah, you receive different opinions. Some of them are happy with this, others prefer something that can be reused if the regeneration is very simple. Yeah.
Yeah, and I mean, just it's fascinating because I mean that really is key to the success of the whole project, really the
Yes.
outcome of it. And I was just wondering, just on that note, I mean, do you or your partners kind of have any kind of get a sense of maybe how kind of stakeholder engagement can be improved, like strategies for ensuring that dairy producers kind of embrace these new technologies?
I think that the work, I mean, this was mainly led by the researchers in Poland. And I think they did a very good job because what I found is that these direct interviews, rather than, they also organize some workshops, but the individual interviews, I think, were very useful. So I guess we received very good feedback. But what is clear is that the next step, we need a prototype that can convince them that the value, as I said, the cost and benefit that is balanced, and that a prototype that can show really that there is a reduction in the mastitis cases in their farms. So I really don't. I mean, I really don't know. I think for that we need probably to work together with manufacturers or I mean, yeah, some stakeholders from industry that can help us to have a first design that can convince farmers, yeah, or can improve the acceptability from farmers.
Sure. Beatrice, thank you so much. I mean, again, I think we could go on and ask so many questions about this wonderful project, but I think unfortunately we'll have to leave it there. It's just past 10 o'clock. I would like to thank once again our excellent two panelists for joining us this morning, Professor Roman Volmer and Professor Beatrice Prieto-Simon, for giving us such wonderful and insightful presentations and providing us with, I suppose, tremendous overviews of the respective projects this morning. The Breakfast Club will be taking a short break, unfortunately, for the summer period. And our next webinar will not be until Thursday, the 11th of September. So I'll be sending out the invitation to all of you after the summer recess. Again, thank you very much to our panelists for joining us this morning, and thank you everyone for attending and for your questions. We really hope you enjoyed the webinar this morning, and we hope to see you again when the webinars resume in September. Thank you very much, everybody, and lovely to meet you both, and have a fantastic morning. Thank you.